生物生産生命環境分野で受賞されました、コーネル大学名誉教授スティーブン・タンクスレー博士にご講演いただきます。
Thank you very much. Can you all hear me? Good, thank you. This evening I'm going to talk about the molecular basis of natural variation and the implications for plant improvement. But I want to put this topic in the context of world food production and world food demand. The human population is continuing to grow, and as it continues to grow, we have a demand for food coming from that growth. But we also have another demand, and that is that humans today are consuming more calories per capita than they were in the past. So the actual food demand is expected to rise faster than the population growth. Now there's a fixed amount of arable or farmable land in the world. So as the human population continues to grow, we actually have less land per capita to produce that food. So if you take all these factors together and look at the projections for the future, it's estimated we'll have to double the yield per hectare of our major crops over the next few decades if we're going to keep pace with world food demand. So that's a very tall order. That's a very large amount of increase that we have to meet in order to meet world food demand. And the question is, can we do it? And if so, how are we going to do it? So in thinking about this problem, it's worth looking in the past to ask, how have we met the world food demand previously? So this graph here shows the yield per hectare of one of the major food crops in the world, which is maize. This is data from the United States taken over about 150 years. And you'll notice the yield per hectare stayed relatively constant until about 1930. And then we see a fairly dramatic rise in the yield at that time. Now this corresponds to the advent of modern plant breeding, and especially the development of hybrid crops. It's been estimated through retrospective studies that about 50% of this rapid yield increase was due to genetic improvement. But the other half had to do with improved agricultural practices, including use of chemical-based fertilizers and pesticides. So in the past, the way we've met the demand is combining genetics with the use of these higher inputs. But if we look at the situation today, we've pretty much saturated the inputs of chemicals that we can put on our crops. And we've also learned from past experience that there's a price for using of these chemicals, that they are not totally innocuous. They can contaminate groundwater and also create risk to humans and other organisms. So we think about the future. If we're going to meet the world food demand in a sustainable way, an even larger proportion of that increase will have to come from genetic improvement, which means plant breeding will have to play an even larger role. So I want to talk about the plant breeding process because I realize at least some of you are not familiar with plant breeding. So this is a very simple schematic. And I'll start by saying that plant breeding is the same as evolution. It's exactly the same thing, only instead of natural selection making the selections, it's humans making the selection. But the process is identical. And it has some very basic components. You start with genetic variation. You exercise selection on that variation. And if you're successful, you have an improved variety. But at the very beginning, at the very foundation of the process is this: genetic diversity. It's the raw material of evolution. If there's no genetic diversity, life would have never evolved. There would be no creation of all the organisms we see on the earth. And if there's not a continuous supply of new genetic variation, plant breeding slows down or stops entirely. So I want to address the question, if we're going to have plant breeding play such a large role, how are we going to supply this genetic variation to fuel the process? So in thinking about this, we can ask the question, where has genetic variation come from in the past to fuel plant breeding? And the answer is that crops, as we think about today, are a relatively new phenomenon on the earth. All crop plants and domesticated animals were taken from wild organisms, wild plants in this case, in the last 10,000 years. So if you look at corn, this nice ear of corn, you think about corn and ask, where did it come from? It actually came from this wild grass called teosinte, which still grows in Latin America. Now this is the equivalent of an ear of corn, which would fit into the palm of your hand. It has very few seeds, and the seeds are almost inedible because they're so hard. Now, like all species, teosinte has a lot of natural genetic variation. The way species survive and adapt to their environment is through genetic variation, which is essentially mutations that have occurred over thousands and sometimes tens of thousands or hundreds of thousands of years. And this reservoir of genetic variation allows the species to survive. So this shows what I cartoon genetic variation shown by boxes. So teosinte had a lot of variation. Humans practice selection on that variation and gradually transform this small thing to a slightly larger, more like corn type, and eventually to the modern corn. Now in the process, a lot of the genetic variation was lost, but some of it managed to come through. So this part that's come through has been the foundation for plant breeding. We're mining this reservoir of diversity still today in the plant breeding process. So if genetic variation or natural diversity is the foundation of plant breeding, we can ask the question, what is the genetic basis of that natural diversity? What actually is behind this process? Now this is a question that relates not only to plants and plant breeding, but really relates to all organisms. Genetic variation is a hallmark of life. So you can see genetic variation, of course, within crops like potato or tomato, but you can appreciate it in our own species. If you look around this room, we're all members of the human species, but every one of us is different from the other in some slight ways, such as height, hair color, eye color, etc. So what actually is causing that genetic variation? Well, this is a question that the founder of modern genetics, or genetics in general, Gregor Mendel, was the first person to develop a technique to try to answer this question. And I'll demonstrate the Mendel's approach to the problem with this very simple, sort of cartoonish version. You can tell I work on tomatoes, so you'll see a lot of tomatoes examples this evening. If I had a very large fruited tomato variety, and I crossed it to a very small fruited, and produced the hybrid, the F1 here, and if I examine the offspring or the children of this, if they fell into discrete classes, like big ones and small ones, they fall into two discrete classes. With Gregor Mendel's principles, you can deduce a single gene was responsible for that difference. So this was a profound discovery by Mendel and opened up the basis of genetics. But the reality is, in life and in nature and in plants and animals and us, very few things behave that way. So for example, in reality, if you take a large fruited tomato and a small tomato and cross them, the offspring don't segregate like this. You get a continuous variation in sizes from small types to intermediate to large. When this occurs, Gregor Mendel's approaches don't work. There's no deduction. If there's no discrete classes, you can't make any deductions. So this was a situation until about 1980. You could answer the question if this was the result. But most of the times, this wasn't what was happening. The variation among us, the variation among crops, is not normally this single gene basis. So we were stuck in this place that we couldn't find out what was the basis of natural variation. So this all began to change in about 1980 with the development of molecular genetic maps. And one of the first maps developed was in tomato, which was done in my laboratory at Cornell University. And this began a process of starting to answer the question, what is the basis of natural variation? So a genetic map is very similar to a road map. On a road map, you may have landmarks, in this case cities. And these cities can be shown along the road. And these landmarks can be used to locate other things. So for example, if someone asks you, is there a McDonald's restaurant somewhere along the road? If it's your children, you say no. But in truth, if it was located between city G and H, you could say, yes, there's a McDonald's between city G and H. So these landmarks provide a way of locating other things. So a genetic map is very similar. You have a chromosome, which is where all the DNA is and all the genes. And you locate landmarks along the chromosomes. But these are not cities. These are called genetic markers. But they serve a very similar purpose. So in this case, these markers become signposts to search for the causes of natural variation. You can use these in a way to search through here and ask, is there anything causing natural variation? And you can start to locate it to some region of the chromosome. So let me demonstrate in a fairly simplified way how this actually works in reality. So again, we have the large tomato and small tomato and everything in between. And we can't do, Mendel's rules don't really work here. But we can take a bit of DNA from each of these plants and examine it with these genetic markers. Now what the markers do is allow you to statistically ask, is there anything around this marker that's affecting fruit size? Now the statistical signal I show here by a little peak here, so let's kind of move along. As we move along this area, this says, no, there's no signal, nothing affecting this, no signal, no signal, and suddenly it starts to rise up, and it peaks the signal, and then it drops back down. So what this tells us is that somewhere around these two signposts, these two markers, is something causing natural variation, in this case, fruit size. And the magnitude of the signal tells you how much it was affecting the trait. So these things began to be called QTLs, which is a genetics nomenclature, but it's an acronym for the word quantitative trait locus, which sounds complicated. But quantitative trait is just another way of saying natural variation, and a locus means position. So what this said is there's a position of something causing natural variation. So continuing on to another chromosome 3, we can move along here. Here's a signal, not quite as strong, but evidence another factor of QTL is located here, and chromosome 8 here. So now we've gone from a case that Mendel's rules don't allow us to study it to suddenly, yes, I know, there's 3 QTLs causing fruit size. This one is larger effect than this one, and these two are smaller effect. So this was a major step forward. It provided an approach for studying natural variation, which is the basis of plant breeding and virtually all of life. So since this time, this method's now been used not only in tomato, but many other crops, plants, animals, and also human genetics to answer the question, how many QTLs affect traits of interest? So in my lab at Cornell, we began to study traits in tomato, especially fruit size traits, to ask the question, actually, how many QTLs cause the variation that we observe? So this is sort of the extremes. This is the actual wild tomato from which tomatoes are domesticated, which still grows in Peru. And this is one of our large beefsteak tomatoes. These are about 500 times difference in weight. What we discovered was that almost all of this difference and everything in between could be attributed to 30 QTLs. We've broken this down and said, yes, we understand there's 30 points in the chromosome, which together can cause all this variation. So this gave a way to start documenting the problem. Yes, there's a certain number of QTLs, and here's how they behave. But it led directly to another question, which is, okay, if I can find in the chromosome what causes natural variation, what exactly happened in the DNA to cause that difference? Now this turns out to be a much more difficult problem, technically, and I'll explain why in the next slide. So I'll give you an example of why this is a difficult problem. So an analogy. So let's say that I gave you a picture of a person named John, and I said, please find John. And you say, well, where is John? And I say, John lives somewhere in the world. Well, at least John is not on Mars. That kind of limits the search a little bit, but not too much. But if I said, okay, actually John on this road map lives in Tokyo. So at least you feel a little better. You're not searching the whole world, you're searching Tokyo, but then you realize, wow, Tokyo is a very large place. You know, how do I find John in Tokyo? It's better than the whole Earth, but it's down to something at least I can drive across. So the analogy is the QTL mapping was equivalent to saying John's in Tokyo. It said, yes, and this chromosome in this region is a cause of natural variation, a QTL. But at the DNA level, these regions can be quite large. They can be hundreds of thousands or even millions of base pairs, which is a lot to search through. So our next goal was to actually see if we could determine the molecular cause of a QTL. And at this moment in time, when we were doing this, no one had isolated the cause. So you're searching for something, you don't really know exactly what it looks like in a very large area. But there were some hypotheses about what causes natural variation. None of these were substantiated because there had not been the work to find out what causes these QTLs. But the three hypotheses were this. If you know about genomes, genomes basically have two categories of things. They have genes, and genes typically encode proteins, and proteins do most of the work in the cells. They cause structure, enzymatic reactions. And we have non-gene regions. Some of those may be regulating genes, some may be doing other things. So the first hypothesis was that what was happening was there were mutations, which I show by a lightning strike here, which caused a change in the DNA of the gene, which changed the codon, which changed the amino acid and the protein. So if you change even a single amino acid in a protein, it can slightly change the properties. It may change the enzymatic properties, maybe a slightly different structure when it folds. But you can imagine that could have a visible effect on an organism. So that's possible. The other was, yeah, it was still genes, but the genes were hit and destroyed. There's many ways to destroy a gene. One is to have a mutation that causes a malfunction in or stop codon, so you get a truncated or an active protein. But you can imagine if you hit the gene and destroy it, that losing a gene could be maybe pretty bad. It could cause something visible that would be natural variation. Now, a third hypothesis was that the genes were not involved at all. The genes were just sitting there. They're doing their job. They're encoding proteins. But what was happening is that the mutations were occurring in the regions around the gene. And these changes were causing changes in how the gene's used, not changes in what it codes for. So for example, a mutation could cause a gene to have a higher level of expression. So maybe a higher level of expression would cause a change in the appearance of an organism. Or it could change the timing of expression. The gene doesn't come on at exactly the right time. It shifts the timing. And even a slight change in the timing, which I'll show in a moment, can have a very visible effect. And the third, it just changes where the gene's expressed. If you take a gene expressed in one tissue and suddenly express it in another, even though it's the same protein, it could have a very profound effect. So a good analogy for this is, we heard a wonderful orchestra last night, so I'm thinking about orchestras and beautiful music, is the orchestra analogy to QTLs. So if we think of an orchestra and the instruments as genes, you can say, well, an orchestra makes a certain sound. How can I change the sound? Well, you could change an instrument. You could substitute one instrument for another, i.e. change gene function, and the orchestra would sound slightly different. Another is you could just remove entire instruments, just pull them out of the orchestra, loss of function, loss of gene function, and that would change the sound. But the other is you don't do anything. You leave the orchestra alone. All the instruments stay in place, and instead, you change the sheet music. Same orchestra, different sheet music, totally different sound. That would be equivalent to changing gene regulation. So again, what we set out to do in tomato as a model system for our work was to actually isolate the cause of a QTL. And we had lots of candidates to work with. As I mentioned, we mapped most of the QTLs for fruit size. And we also did fruit shape and some other traits I'll talk about. I'm not going to go over all the details of how we did it, because that would take too long and probably put everyone to sleep, including me. But I'm going to give a few examples to show technically how we did it, but also what we learned from it. And the first example I want to give has to do with mating systems. So like animals, plants are capable of mating with each other. But unlike animals, plants are also capable of mating with themselves, which is probably why plants never get lonely. Sorry, that's a very bad joke, but it's a plant humor. But there's several factors that determine whether a plant mates with other plants or mates with itself. And one of the key factors is the floral structure, the structure of the flowers. So this is a tomato flower. If you see it in your garden, it looks like this. If you cut a cross section, you'll see both the male and female parts. So this is the male part. This is the anthers that produces the pollen. This is the female part. This is the style. This is the stigma. The pollen lands here, it grows down here and fertilizes the egg. Now in this case, this stigma, which receives the pollen, is sitting inside the flower, so it's not accessible to pollen from other plants. It's closed for business. So the pollen's released, then it fertilizes itself. So the cultivated tomato is self-pollinating, doesn't need anybody else. But other tomato species and many other plants are cross-pollinating. They're outcrossing to other individuals. And here's an example of an outcrossing tomato species flower. Here you can see the female parts, especially the stigma, is exerted. It's sticking out. So it's available for pollen from other plants. And this is aided by bees. The bees, this is a bumblebee, will attach to the flower and vibrate. The pollen will fall out, and then we'll carry it to the next flower. So we were interested in what is determining this difference in this length of this style and stigma exertion that might be changing mating system. So we used the QTL mapping approach I already outlined, and what we found was there were shown in blue here a number of different QTLs that affect how much this sticks out or does not stick out. And we chose one of them, which we called SE2.1, because it's on chromosome 2. SE stands for stigma exertion, and decided to try to pin down exactly what had happened in the DNA. So again, this was a big challenge because even we knew this QTL SE2.1 is on chromosome 2 in this region. When you blow it up and look at the DNA, there's actually lots of genes in the region. These are genes in red here. So there's about 20 or more genes in here, any one of which could have been affected to cause the QTL. So it's still too large of search space. So what we did was to use a genetic technique which is called fine mapping. What we did is collect systematically crossover points in the chromosome shown by X's, and these became additional landmarks for locating the cause of the QTL. So here's a bunch of X's, and our experiments indicated the cause of the QTL actually existed between here and here. Well, the search space is reduced. There's now just 3 genes in there, but that's still a lot of space to search. So we pressed on further, and after a lot of work, we found a few more recombinations, one here and one here. And this is where the QTL was. And when we examined it, to our surprise, there were no genes. So this would be equivalent to knowing that John lives in Tokyo, have worked very hard, and found the address of John. You go to John's house and open the door, and no one's living there. So again, a QTL had not been isolated, so we didn't really know what to make of this. But we did have a hypothesis. There are no genes in here, but there's a gene sitting over here just outside the interval. And just upstream of the gene is this region. So a hypothesis was maybe some mutations that occurred in here that changed the regulation of this gene. So we sequenced that region from both the long style plants and short style plants to see if there were any mutations differing between these two. We found there are many, many mutations. There was These arrows indicate base substitutions. These triangles indicate insertion deletions. So yeah, there was things that could be causal, but there was a big dilemma. If you have a mutation in a gene, you can pretty much predict what it will do because of the universal genetic code. If it hits this, changes that, it changes a codon. But there is no universal code for regulatory regions. We have still, at this moment in time, fairly limited knowledge of what actually causes regulatory changes, which meant that any of these could be causing the difference or none of them. So the next thing we had to do was demonstrate that, yes, the cause was in this region, even though we don't know what it is. So we used the method that was available in plants, and it's still available in plants. We took the regulatory region from a long style plant, and we added it back to the genome of a short style plant. Now, if the regulatory factor was causing the style length, then this regulatory region should cause long styles. This is exactly what we saw. So this proved that the cause was upstream of this gene. We further went on to discover the function of this gene. This gene encodes a protein that regulates the cell elongation, how long the cells get. And it's very precise. And this is what is, to me, was pretty marvelous. This is a very blown up section of the style, which is the female part of the plant. But in reality, it's just a few millimeters through here. This gene is expressed in this one part of the style. And in the plant with short styles, it's expressed just a little bit less. And the one with long styles, a little bit more. But that expression is enough to cause it to stick out more and produce this. This is pretty incredible. It was a change not in the gene itself, but a change in the regulatory region for a gene that's expressed in a very small piece of the organism. And just a small change in the amount was enough to change the mating system of the plant. Second example I'll give was isolation of a gene, isolation of QTL, or finding the cause of a QTL involved in fruit size. So again, you have fruit sizes here, and we've done QTL mapping. We chose one of these to study. We did the same kind of experiments, and lo and behold, we got the same answer. The gene itself had not changed at all, but there had been a mutation in the regulatory region. And in this case, the gene encodes a protein that controls cell division. And the number of cells that accumulate in a developing fruit, you can imagine, will change the fruit size. But the change was a little different than the previous example. It slightly changed the timing of expression and also changed the amount of expression. So it was a change in timing and also a change in the level. In a fairly short period of time, we're talking just about a few days, but these shifts were sufficient to change the number of cells that accumulate to go from smaller fruit to larger fruit. So this is a second example where it was not a change in the gene itself. The third example, which is a QTL that affects fruit shape. Tomatoes, like most fruit, can take different shapes. You can have nice round tomatoes or pear-shaped tomatoes. So we found a QTL involved in changing shape from round to pear shape. And in this one, when we found the cause, it actually was a cause in the gene. It was a mutation that caused a loss of gene function. So you eliminate that gene, which apparently is involved in causing shape, and you switch from one to the other. So the upshot from our work, and this are just three of the cases that I mentioned here, we had none of these examples that were changes in the gene. We had none that were changes in function. We had one that was loss of function in the gene. We had two that were regulatory changes. So time has elapsed since these studies were done, and now the similar approaches have been used to isolate other QTLs in tomato, but also in a wide variety of plants and even animals. So I went to the literature, and this is not comprehensive, but took a sample of where people have found the causes from rice, tomato, maize, and Arabidopsis. And actually, all three categories are participating. We're having a change in gene function, loss of gene function, but a disproportionate amount is change in expression, not changing the genes at all. So going back to the orchestra, it appears that a large portion of the variation that we observe in nature and also plant breeding is caused by changes in the sheet music or changes in gene expression. So this brings up a question. I pose this whole thing in the context of where are we going to find the genetic variation to fuel plant breeding? And a sub-question would be if by looking at what Q-tails are, we find out that a large portion are caused by changes in gene expression, where are we going to continuously provide or find this source of variation to keep the plant breeding process moving forward? So today, and I'm sure this will change as technology moves forward, we really have three approaches to provide this variation. One is GMO transgenes, other mutagenesis genome editing, and the third is untapped natural genetic diversity. Now, all of these methods have exactly the same goal, and that goal is to supply a continuous set of new genetic variation to the gene pool to fuel plant breeding. They have exactly that objective. And I want to make sort of a comment here just in terms of us as society, how we view what we're doing to feed ourselves and plant breeding in general. You can ask yourself, are any of these technologies good or bad? The technologies themselves are neither good nor bad. What can be judged good or bad or in between is the outcomes of technologies. What have you done with this? So, for example, you can use natural variation to improve yield, but you can also take natural variation, which occurs in wild plants, and introduce toxic compounds. A good analogy to this would be the pharmaceutical industry. You know, the pharmaceutical industry makes drugs, right? And they have many techniques for making drugs. They don't use just one technique. They have a whole battery of techniques to produce the drugs. And then those drugs undergo testing to see if they have efficacy to treat human diseases. And they also have side effects, which may be even worse than the treatment. If society judges that the benefits outweigh the risks, they're released to society, and society gets to use those drugs. But when a new pharmaceutical comes on the marketplace, no one asks, which technique did the pharmaceutical industry use to make that drug? They ask, what is the benefit or risk of that drug? So the same thing applies here. I think as society, if we're going to meet the needs for feeding the world, we need to focus on, are the outcomes of the technology beneficial or not in terms of feeding the world and creating risk for health or not, versus are technologies good or bad? Because technologies are neither good nor bad. So I'm going to go over each of these technologies briefly just to give you an idea of what they do. Mutagenesis and genome editing. So whenever people hear the word mutagenesis, including myself in the past, was like, mutations sound horrible. I mean, and many mutations are. But the reality is mutations are the basis of life. If there were no mutations, there would be no life on this planet. There would be no diversity, and we would not be here, and there would be no plant breeding. Inherent in DNA, it's an unstable molecule, is to mutate. And on top of that, there's natural things in the environment, like radiation, that are creating mutations. So we knew in the past, for a long time, mutations were occurring naturally. Like I said, it's part of life. But we didn't know how they were occurring. And it wasn't until the mid part of the last century that people first discovered some of the things that cause mutation. The Nobel Prize was awarded in 1953 for the first discovery of what causes mutations, and this was ionizing radiation. And that discovery was a basic discovery, but also created a great enthusiasm in plant breeding, because in the past, if the genetic variation had not occurred, you couldn't use it. You could say, well, maybe I could wait around for it to occur, but it could be 1,000 years or more. Said, no, we'll just make it happen. So there's a big euphoria. There's a big period of about 20, 30 years when people are applying mutagens to plants, trying to find useful variation. And the results were not very beneficial. And the problem was is you couldn't specifically mutate anything. You could just shoot the mutation at the genome, and you mutate many things. You hope one was useful, and usually it was not. And even if you got a gene that was useful, you had another 50 mutations in the genome that weren't. So by and large, the technique had very little impact on plant breeding. We have new technologies today that allow you to target mutations to specific parts of the genome or specific genes, and this is called genome editing. Without going into the details, you have a vector that specifies a particular piece in the DNA that you want to cause a mutation, and moreover, you specify which mutation. And by doing this, you can go into a certain part of the genome and make a change, a very specific change. So this has overcome two of the problems with mutation breeding. One, it's specific. You don't have to mutate anything else, and you can cause a mutation, the exact mutation you want. Now the limitations are you need to know what gene to mutate and what kind of mutation to make. Now there are things in plants that are important to plants. We know which genes are important, and we can use a technology to create the beneficial mutations. But there's still a big knowledge gap. For many things like yield, we don't really know which genes are important for yield in most crops. And if we did, we don't know exactly what mutations do occur. And moreover, as I already mentioned, many of these are regulatory changes. We have very little knowledge about how to make regulatory changes. So the upshot is this technology has some applications now. I think as our knowledge base increases, this technology will play more and more of a role in providing the useful variation to fuel plant breeding. Another one is GMO transgenes. And this has an approach that I don't have a gene I need, so I'll add a gene to the organism to make something that I find useful. And a good example is the Bt protein. The Bt protein is naturally produced by many microorganisms, or certain microorganisms. And it was found long ago that this protein, while it's innocuous to humans, it's a protein we eat like any other protein if you want. For certain insects, it disrupts their life cycle. It's toxic. But it's not toxic in the environment. It's a protein like anything else. So it was one of the very first organic pesticides used. It's still used today as an organic pesticide. But the way it was used, you had to grow the bacteria, harvest the bacteria, spray them on the plant. Insect eat the plants, it dies. But the plants don't have the gene. So what was done was just put the gene in the plant, make Bt, for example, cotton. So the plant's producing its own proteins, so you don't need to go and spray it onto the plant. Now this has had a remarkable impact on in a number of ways. First, it's for crops that have high insect pressures, it's increased yield. But it's also, and cotton is a very good example, cotton in the past has consumed more pesticides than any other crop. I mean, huge amounts of pesticides to protect it. Bt cotton, which is now grown widely, has significantly reduced the amount of pesticides, chemical pesticides required to grow this crop. The limitations on this technology is there's not a whole lot of genes we know that we can add to a plant that would help it. There are a handful of genes, but it's very limited. So there was some initial successes, but it sort of had this lag in that our knowledge base is not sufficient. What will happen in the future, I don't know. I'm supposing that methods like genome editing may actually become the predominant method on top of natural variation I'll talk about. But GMO transgenes do have a role. The third area I'll talk about, and this is where most of my career has been spent, this is untapped natural genetic diversity. So as I mentioned, all species have this natural variation that's accumulated through natural mutations over hundreds, sometimes millions of years. And we realized this long ago as a society and began collecting the natural variation in seed banks. So for example, today, if you're working on wheat, there are 37 major seed bank collections with about 400,000 seed types available. Moreover, as a global community, we've taken the efforts to preserve this for the long term. There's an island, a Norwegian island near the North Pole, where they've dug into the mountain and made a vault in here to store seeds. This is called the Svalbard International Seed Vault. And the vault aims to safeguard the world's agriculture from future catastrophes such as nuclear war, asteroid strikes, and climate change. I hope they don't all occur, although we are in the latter one currently. The facility has a capacity for 4.5 billion seeds. So to meet the world's agricultural needs in the future, it will be important that we continue to collect and preserve these seed banks. This is the natural heritage of our biological species that takes a long time to accumulate. But the issue really today is how to use this, how to make more efficient use of these natural resources in these banks. Now the problem with doing this is twofold, because if you look at these seed banks and ask how many of those 400,000 wheat seed types that have actually been used for wheat breeding, it's probably less than maybe 4,000. Just a small portion ever get used, not because they're not potentially useful, but because it's very, very expensive to use them. With the current methods, we can only sample a fairly small number of these to put into breeding programs. And the other is that even if we sample them, it's not always obvious by looking at them if they're useful. For example, teosinte, the progenitor of corn, still has useful genes that can increase the yield of maize. It's been demonstrated, but you can't see it by looking at it. You have to actually do some breeding work to discover it. So the challenge today is just making better use of our gene banks. And fortunately, we're in a period of time in life sciences that there are significant advances that can be made and are being made to improve this situation. And this has been called in general the genomics revolution, which is actually two revolutions. The first revolution was how do you create huge amounts of precise genetic information, i.e., how do you sequence on large scale with high precision at a low cost? This part of the revolution took place, it's still taking place, but a large part took place over the last 2 decades. And this was a purview of chemistry and engineers. The chemists built the chemistry to do it, the engineers built the machines to do it. And as I'll show in the next slide, it's been very, very successful. But what this has created is a vast amount of information that needs to be used. And the use is coming from a different field, mathematical algorithms. And this is coming from the fields of mathematics and computer science. We're just into this phase of the genomics revolution. But just to give you an example of how well the first period of the revolution worked, the human genome, the first human genome was sequenced in 2003. So one human genome. It cost $2.7 billion for that one genome. Now, a lot of that went into technology development because a lot of the technologies didn't exist, but it still cost $2.7 billion. 2005, 2 years later, the next primate genome was sequenced, chimpanzee, came down to the very low price of $100 million. 2010, you could do it for $100,000. Last year, $1,000, complete sequencing of your genome for $1,000. Today, we can do methods that are called next-gen sequencing, which are essentially sequencing the majority of the information containing content of the genome for $40. Not just for humans, but for any organism, any plant, any organism, you can get the genomic information for $40. So you can see this curve. This is even more impressive than Moore's law. And you can see in the future, this is going to be a very nominal, a very small marginal cost. So coming back to the breeding process, breeding is no longer limited by the availability of information. The key is how to use information to make better decisions throughout the breeding process. So for those of you, if there's students out there or anyone else that has an interest in mathematics, computer science, operations research, I would encourage you to consider a career in life sciences and even plant breeding, because it's you. You will be the people that create the next revolution, the next phase of the revolution in life sciences and plant breeding. Now, on a personal note, I've made my own effort to try to facilitate this in plant breeding. In 2006, we formed a company called Nature Source Genetics. And it's a company devoted to increasing the efficiency of plant breeding through merging the mathematical, computational, and quantitative scientists. So we hired people from basic mathematics, operations research, computer science, gave them the vocabulary to work on plant breeding problems, and have been developing mathematical algorithms for the plant breeding process. And the last couple of slides, looking at Toshi here, he's got a sign held up here. I watch Toshi. He's my minder, don't want to cross him. I'll show you a couple of examples of how it works. So as I said, one of the challenges of using natural variation, there's way too many things to use. How do you sample what to use? So if you have 400,000 wheat lines available, and you can only practically breed with 100, how do you choose the 100? So I'll give us an example. It's a smaller version of it, but it's the same principle. Let's say you only had 200 lines, and you could work with 20. One subset of 20 is the best to work with, only one. Question is, how do you find the best 20? Well, let's look at the numbers. You can just do it by chance. Your chances are not very high. With 200, choose 20. There's this number. It would take more than 5 minutes to say, so I won't do it. It's more than the stars in the entire universe, but only one is optimal. So we've been doing it at NSG, is developing algorithms that use both sequence data and other data to solve this problem. So we're able to find the solution for 200 choose 20, or even 200,000 choose 20, to help solve the sampling problem. So this is the first part of the problem. How do you sample? And this is where optimization and genomics data can significantly help the process. The other is, once you get it, how can you find the usefulness of it? Because it's not always obvious. We developed another technologies called optimized discovery populations, which I won't go into, but it essentially converts material to finding what's hidden useful genetic variation. And if you string these together, the process we've developed can allow a steady supply of new and useful genetic variation to fuel plant breeding. So we've been at this for a number of years now, and we've refined and are continuing to develop our technology. But I'll give you one example of where we've used it. This is it's a crop plant where currently the results from all breeding worldwide, all companies, is about 1% gain in yield per year. Using the methods I just described, the optimal sampling and optimized discovery populations, we've been able to take the original material and increase the yield by 19%. So you can see this as a big jump in efficiency. So the implications is we can make better use of natural variation through the role of optimization. And we've tested this both in vegetable and field crops with similar results. So I just want to end up, I don't want to be a pessimist, because I really do believe we can meet the world food demand. It's not going to be just one thing that does it. Plant breeding is going to have to play a major role. But there's no single technology in plant breeding that's going to accomplish this. So I think there's two responsibilities here. One is the responsibility of people like me, working in the field to do our best to bring the best technologies to the problem. But the other responsibility is a society's responsibility. All of us have a vested interest in feeding the world. If we don't meet the world food demand, the problems we have now are going to be minor in comparison. To do that, we need to understand it as a general public, the basic breeding process and how to make decisions. We can make decisions about how to use technology usefully, but we have to be able to do this in a way that actually moves us forward. I think if we do this, we can meet this demand and feed the world in the future. So with that, I'm going to conclude, and I want to thank a host of people. I won't list them I won't say all their names, but both at Cornell, which is where I did the majority of my research in the past, and Nature Source Genetics, where I'm spending the majority of my time currently. And with that, I thank you very much.